Dr. Unal. I am a scientist from Turkey, and I'm the Secretary General of ISAC, International Society of Antimicrobial Chemotherapy. I am welcoming you all in the name of ISAC to this webinar program that is the part of ISAC Academy, a newly established training program. And we have been doing that for the last 1 year, and we are doing webinars on important subjects. And today we will talk about the pandemic of the century, the coronavirus, COVID-19, and about the vaccination. Actually, at the beginning of the pandemic, as a part of our webinars, we have done the program about vaccines again. From the first one, at that stage, the early vaccines were still being developed. Now we're at the stage of revaccination and variant vaccines. And the subject of the webinar is this: revaccination, is it necessary? And what are the variant vaccines? I would like to make a short introduction to the webinar. All the things that you are very aware of. You know, dear colleagues, these new model outbreaks, mainly for the viral ones, are seen at the different part of the world, like the Ebola virus, Lassa fever, MERS-CoV, SARS-CoV-1, Rift Valley, Zika, and we all were expecting the newcomers because of the globalization of the world, global warming, and the better transportation, all that stuff that we have are aware of. And at the end of 2019, the COVID-19 became the new one. And you know the story, starting from Wuhan, starting from bats to the intermediate animal, and finally to man, and all over the world. And with the waves, we suffered with the waves, different variants of the virus. We dealt with the 620 million total cases, and there were 6.5 million deaths. And still, as you see over here, there are still new ones still going on. The death rates are going down. It's okay, very good, but the pandemic is still going on, and we have to keep our fighting against that worldwide problem. And the best thing we have done against that, of course, to protect ourselves, we learned wearing a mask. Mask saves lives. Clean your hands, keep a safe distance, and we learned how to diagnose it with the PCR sets and with the antibodies. And the treatment plan, we all have memorized that slide all over the world. Early stage antiviral agents like the remdesivir, molnupiravir, and the Paxlovid, but before that, world has used hydroxychloroquine, favipiravir that were not very effective. And we learned about the oxygen therapy, anti-inflammatory therapies, mainly the dexamethasone and the other JAK inhibitors, monoclonal antibodies, fighting against the host immune response. As all those things, we all fight against that. Of course, the mainstay of the fight was the vaccine. And the people who are dealing in the field of vaccine, we knew that the methodology was quite complicated. We have old ones, we have new ones, but each of them necessitates tens of years. But at this stage, the forests were burning. I mean, burn, fire was every place. So we could find a definite solution. And thanks to God, in less than 1 year, the world could develop the vaccine. And remembering the similar ones, 1918, influenza pandemia, the death bodies were much higher. And the major difference in between two is the vaccine. World could develop vaccine very fast. They were inactivated whole virus vaccines, but we have been introduced to the nucleic acid vaccines, particularly the RNA vaccines, and we have been introduced to viral vector vaccines and some protein-based vaccines. All that different platforms in the different part of the world. Hundreds of vaccines has been developed, but it's not the end of the story. Developing vaccine is not solving every problem. You have to upscale it, produce in millions, and thanks to God, in different kinds of vaccines, the world could generate, I wouldn't say enough, but quite high number of the vaccines, and with the help of the vaccine, we could slow down the number of the number of the deaths. Of course, it was very valuable, but at the same time, we were suffering from the problem of variants. Since there are so many multiplying viruses, the variants were being chosen among them. We have started with alpha at the end of 2020, and then the beta and gamma was not very effective, and particularly delta, and finally Omicron. So the variants question over here today is, are our vaccines, that is available vaccines, effective to these variants? Do we need variant vaccines? So today's meeting, we plan to talk about this subject. First question is, how successful was the vaccines? And at this stage of the outbreak, do we need revaccination? And then second questions, what kind of vaccine do we have to use? And we will discuss this subject with 4 distinguished scientists who are involved in the subjects deeply. I'll introduce you each speaker before they start to begin their lecture. And we will start with Dr. Rafael Araos. Dr. Araos is a research professor at the Institute of Science and Innovation in Medicine, Universidad del Desarrollo, Chile. Dr. Araos is a physician scientist trained in internal medicine and infectious disease. He is interested in understanding the transmission dynamics of infectious disease and evaluating the impact of health interventions. And during the COVID-19 pandemic, he heavily involved in the subject, and his group built a popular-based electronic cohort that has allowed them to determine the COVID-19 vaccine's effectiveness in the real-world setting. Yes, Dr. Araos, the scene is yours now.
Hello, everyone. Thank you, Professor Unal, for inviting me to present today. It's always challenging to keep presentations up to date. So I tried to include new data. So I think there's a bonus track at the end, just to show and tell and make the discussion more fun. So the idea, right, is to discuss about revaccination, considering all the vaccines that we have available worldwide, and considering also the data that support that use. So my name is Rafael Araos. I work in Chile, and my team, it's been trying to assess vaccine effectiveness in the real world setting since almost 2 years now, and that's going to be my perspective. So I'm not going to be very molecular. My presentation is more related to observational studies. So I have several conflicts of interest, mainly with AstraZeneca. I've been speaker a couple of times and I've participated in advisory boards. Now I have to include Sinovac, and I have research support from different entities, and also I participate in some boards for evaluating grants. So this is the epidemic curve of the COVID pandemics. You know it very well. First wave, then we have in several parts of the world P1, then Delta, and then we have these huge Omicron in different stages, right? Original and then sublineages, and this is happening even though we have vaccines. So we know that at least from a case perspective, there's a need for doing something if we want to stop transmission. There's a huge discussion about that. It was one of the aims at the beginning of the vaccine era of the pandemic to stop transmission, but now we think that it's more important, right, to avoid severe disease. But anyways, we still have to see how new vaccines behave in terms of avoiding transmission. And this wouldn't be a terrible problem if cases wouldn't be translated into death. And even though we know that Omicron and the related variants are less aggressive than prior ones, we still see a significant number of deaths, right? As you can see here, this is the daily number of deaths. Again, was pretty related to initial vaccines, a lot of impact, initial variants, sorry, a lot of impact, and then with Omicron we saw less death, but still a significant number that goes up with every outbreak. So we still have morbidity and mortality associated with Omicron. This is another way to see this. This is a mortality excess estimation that The Economist always published. This is from another web page, but the data is from their predictions. And as you can see here, this is the estimate of excess mortality starting, right after the first cases of the Omicron of the COVID pandemic started. And as you can see here, even though it is going down, it is still a significant number of deaths that are above what we were expecting to have without COVID. And some of those excess mortality is explained actually by confirmed COVID death, but there's still a good number of unknown cases. There's probably some COVID and other indirect causes. But the thing is, even though we have, vaccines, very good vaccines, unexpectedly good vaccines, we still see a significant mortality associated with the pandemic, and that seems far from returning to normal. That's, for the way I see it, and many people see this, it's a red flag and, a challenge to everyone in terms of how we are going to face this in the near future and try to make more efforts to decrease the excess mortality. And of course, one alternative is to continue and to improve our vaccination strategies. So this is where we are standing now in terms of the genomic landscapes. We're in the middle, let's say, because there's no signs for the Omicron wave to end. We're facing different subvariants or sublineages of the Omicron variant that emerged from different pathway compared to other variants. So it's a new one, right? And from the antigenic composition is pretty different. You can see here, this is a very nice cartography, antigenic cartography. It's a preprint, but it's a really fine paper that you can look at. And you will see how this is like a PCA. So the closer 2 points are, the more similar they are. So you can see here that all the prior variants tend to cluster together, and Omicron cluster really far away. So that's telling us that from the antigenic perspective, Omicron is pretty different than the other ones. And then when we analyze the immune response against vaccines, not natural infection, we also see that the coverage of the current vaccines are super close to the older variants, but Omicron, still, stands far from the variants. And that's telling us that current vaccines may have a problem for controlling infection at least. So taking that into consideration, that we still have a relevant variant that still causes morbidity and mortality, and that's super different from prior variants. These subject, the vaccine waning, the immunity waning, becomes super relevant, because we knew that before Omicron, when we analyzed in this paper that led Daniel Faking, when we analyzed the trajectories of the immune response against what was circulating at the beginning of the pandemic when everyone was receiving primary series, we learned that immunity waned, right? And this is several examples of the effectiveness of different vaccines, mainly mRNA and viral vector vaccines, over time after the primary series was completed. And you can see that for every outcome there was a decline in the protection, and that was before Omicron. So we already knew that immunity waned over time. And that was even worse when we were including papers with the Delta variants. So that's one thing. So knowing that protection was not forever after the primary series, countries started to think about using boosters, right? And that was before the Omicron era. So many countries started using booster, different approaches. Some were using homologous mRNA boosters. We learned very soon that it was a good idea. Then other countries started to use heterologous boosters, mRNA above inactivated vaccines, viral vector above inactivated vaccine, all kind of combinations. And data started to emerge. And we learned from the UK group, from Dr. Lopez Bernal, that ChAdOx1, so vaccine from AstraZeneca, and Pfizer's vaccine was super effective for improving, vaccine effectiveness as a first booster. You can see here that after boosting a primary series with, in this case, Pfizer's over ChAdOx primary series, we reached like 100% effectiveness against Delta. And then when we did Pfizer's above Pfizer, you can see that the effectiveness that was reached was also super nice. And then when we look at what happened against Omicron here in these bars, you can see that although the improvement was less than for Delta, was still good. So this was one of the first papers showing that a first booster of, in this case, mRNA vaccines over different or on top of different primary series was a good idea and a good intervention for any of the variants that were circulating, in this case in UK. So, and they use it like 6 months after having completed the first, the primary series. That's one thing. So then we, at the same time, we were trying to see in Chile what was happening here. We had a very particular landscape of primary series because we used primary series of CoronaVac, which is an inactivated vaccine. We used AstraZeneca, we used mRNA vaccines, we used CanSino, which is another vaccine from China, and we also used some combinations of Corona or mRNA vaccines with AstraZeneca. So the possibilities in the country were pretty a lot, and it was a challenge to try to understand what we were seeing. So we focused on what was particular from our country compared to what England was doing and what Israel was doing, which is basically mRNA boosters. So we followed our cohort, which is an administrative cohort that's countrywide. So everyone that's in the public health system, is 80% of the population in the country, is covered by this cohort. And we merged data sets that are built up in real time, so we can have information regarding exposure. We know exactly when, what, and the number of vaccines that every citizen in the country received. So we know the exposure, and then we can, over time, capture incident cases of COVID, either asymptomatic or symptomatic, or we can know exactly when subjects go to the hospitals, whether they are hospitalized or not, whether they are transferred to ICU or not, and whether they died or not, and the cause of the death. So we can have the exposures and we have the outcomes, so we can estimate vaccine effectiveness, and we can adjust that for several covariants that are important for trying to isolate the effect of the vaccine. And when we do this, you can see this is not a very beautiful slide, super busy, but let's just focus on the right column when you have the stratified, weighted, adjusted analysis. So these are the numbers that are final for us. And then on the left, you have the groups. This is the outcomes. So this is infection, no matter whether you are symptomatic or not. This is hospitalization, ICU admission, and death. And then you have all the possibilities that we used in the country. So here we are analyzing the effect of CoronaVac, BNT, Pfizer's, and Astra vaccines as boosters on top of a primary series of inactivated vaccines. So it's that was pretty unique at the time, because no data was being published. So what we found was any booster actually was pretty good for increasing protection against all of the outcomes. As you can see here, CoronaVac went to the primary series of CoronaVac in the best moment was 67-68% effective against infection, and the first booster, homologous booster, right? In this case, 3 CoronaVac vaccines. The booster was put like 6 months after the primary series was completed, was almost 80%, and you can see the numbers for the mRNA and the Astra booster, almost 95%, with very, very narrow confidence intervals. The same for hospitalization. Here you can see CoronaVac reached almost 85%. That was also better than the primary series, and the other 2 options that were heterologous boosters with BNT or with Astra was close to 95-97%, and that's the same for ICU admission and for death. So we learned here that no matter what kind of primary series you had, and no matter the booster that you were using, it was a good idea to start putting third doses after primary series of CoronaVac having completed 6 months after the last dose. And this, was a confirmation of the Israeli data that were saying that a third dose of mRNA on top of mRNA primary series were, super nice. And that's the same for the UK data. This was during the Delta period, so no Omicron here. And that's, the challenge that we're facing now. So what happened? This is the epidemic curve of my country, of Chile, and you can see that we have this is our first wave. Then we have P1 here. Then we started the booster campaign around here. So the third dose was rolled out in Chile before the arriving of Delta. Then we have a Delta wave that was pretty mild compared to what happened in other countries. And again, we think that it was because of the most of the vulnerable population was already vaccinated with a booster. And actually deaths were also significantly lower than in other countries. But then, like 6 months so after having used the having rolled out the third doses, we had our first cases of Omicron, and then we had a huge outbreak like everyone else, but our outbreak was pretty high, and we also saw more death than what we were expecting to see. And so basically seeing what was happening at that time regarding the dynamic of the death associated to COVID, we decided to go for a second booster that we started to roll out by the beginning of February this year. So in the middle of the Omicron wave. And then we had these B4 and B5 outbreaks. So, so now the idea is to try to understand whether, second doses are important or not. So we already know that third doses or first boosters, the way you want to call it, are a good idea during the Delta period. The UK data showed us that third doses were also a good idea during the Omicron period in the paper I showed you at the very beginning. And now we're going to see if a fourth dose or a second booster during the Omicron wave, it is also a good idea or not. So this is the dynamic of the genomic, from the genomic perspective. You know that during the first half of the pandemic, there was a pretty, so waves or variants were not very coordinated. For example, in South America, we have a significant outbreak of gamma, but we almost didn't see beta, etc. So it was different until Delta came, and then Omicron, in which everyone synchronized. That's probably because of how transmissible it is. So now basically all the world is dominated by Omicron in different waves, and we are now facing pretty much everyone a BA5 and other subtypes of BA5 outbreaks. So what we want to know is whether the third doses work for the right tail of the table. And here you can see a first paper. This is the group from Israel that probably is the group that leads us leads the production of information regarding boosters, because they are always very, very rapid in let us know what's happening, and that's great. And as you can see here, during the Omicron period, a second booster of Pfizer that was mainly for people at risk. So this is very important data. It's not just healthy population, it's people that have more problems dealing with the disease than others. They showed that the cumulative incidence, and when they showed this in terms of relative risk, was significantly lower for the fourth dose group in every outcome they were following. So that's first news, right? So it seems that a fourth dose of mRNA vaccines on top of a 3 mRNAs background was a good idea for infection, disease, and severe disease when you use the fourth dose. I think it was like 4 to 5 months after having completed the primary series. And then there are several data. My group is working, and I'm going to show you that, because again, we have a very particular matrix of vaccines. So it's always nice to see if we can, in a way, replicate what Israel is seeing for homologous mRNA boosters. So this is specifically what we are seeing in Chile since February this year. So this is when we started putting second doses. We mostly put second boosters. Sorry, we mostly put second boosters of mRNA. Like 98% of the second boosters that we have used are mRNA vaccines. So this is in a way an interrogation against mRNA vaccines on top of different 3 doses backgrounds. And we are so what we included in this period is basically BA1, then BA2, BA4, and BA5, which is the last part of the plot. So we are when we look at the data that I'm going to show you, we're considering only Omicron and Omicron sublineages. So sorry, this is in Spanish, but this is just the unique. This is just the title, so don't worry. And we have a cohort, as I said, that allow us to consider different exposures and different outcomes. Now we are going to be analyzing 4th doses of mRNA on top of different primary series that I'm going to show you, the most common ones. And we are going to be analyzing the effectiveness of this booster against ICU admission and death. We're not going to be, we're not focused now on infection and COVID because with so many cases, we think that our outcome of infection is less robust than it used to be. So we are focusing on what we are more sure about, the classification of the outcome, which is ICU admission and death. So this is just to show you the general numbers. The cohort, when we exclude people, you will be able to see this very soon in a preprint. But basically, we have almost 4 million people that receive 3 doses of any vaccines, and that received the last dose at least, the last dose, so the 3rd dose at least 24 weeks before the beginning of the study. And then we compare those that received the 4th dose against those that are not vaccinated. So what I'm going to show you now is how people with 4 doses do compared to those that are not vaccinated. So I'm not considering here 3 or 2 doses alone. I don't want to compare 4 versus 3. Just want to tell you how people with 4 doses do compare with those that are still unvaccinated. So it's 2.5 million people against almost 3/4 of a million people that are still unvaccinated in the country. So this is just demographics. So this is how the rollout went. As you can see here, in the group that we analyzed, we had an incredible uptake of the 1st doses, and the 2nd doses were almost a mirror. So most people, follow the schedule as they should. Then we have the way they received the 3rd dose. Probably, the first half of this cohort, which is the elderly population, was very, very strict, and they received the 3rd dose as they should. And then the younger people were, the slower they started to receive the 3rd doses. And then you can see here how the dynamic of the 4th dose was against for the elderly, which is this group. It was pretty rapid, and then it was slower, which is something that we were expecting. Again, the number of people that received a 4th dose is pretty important, and as you can see here, in a period of from February to mid or to the beginning of May, more than 1.8 million people already received the 4th dose. And then you can see effectiveness. So this is new. It's going to be published very soon. And the idea is to show you 4th doses against unvaccinated people. So here you have, so in the upper part of each slide, you can see the combination. So on the left, you're going to see vaccine effectiveness against ICU admission in general. So any primary series plus any booster, 2 boosters, and death, any primary series and any booster, right? So this is just to have an idea about how well a 4th dose work in general. And as you can see here, works pretty well. So 6 months, a bit more after having initiated the rollout of the 4th dose, more than 80%, or actually the vaccine effectiveness is close to 82, 85% when you look at 4th doses in general against ICU admission. And then for death, you can see the same. It's a bit better. It's around 85, 87% effective against death 6 months after the beginning of the 4th dose rollout. As you can see, there's a significant decline in effectiveness, but in terms of absolute numbers, this is pretty unimportant. And then just to show and tell, this is what we have, what you can see when you look at the specific regimens for ICU admission. It's pretty much the same for death. First, we're going to see what happened when you use the primary series of 2 mRNA plus 2 mRNA boosters, and you see this drop in effectiveness, which is caused by 4 cases. So again, looks not so good, but the numbers are so few that we have tremendous confidence intervals. And as you can see, more than 6 months after the initiation of the 4th dose campaign, we have more than 75% protection against ICU admission for all the homologous mRNA boosters. And then here you can see, so this is a primary series of inactivated vaccines plus 1 inactivated booster plus 1 mRNA booster. So this is a very particular 3 coronavac doses and a Pfizer or Moderna booster. As you can see here, it's pretty well against ICU admission. 6 months after the initiation of the rollout, you still have more than 75% protection against ICU admission. Then we have here 2 inactivated, so 2 Corona, 1 Astra, and 1 mRNA booster. This is a very interesting group because most people here is people above 75 years old and with comorbidities. So having this curve 6 months after the beginning of the 4th dose period, it's super good news. We're close to 80% effectiveness against ICU admission with very narrow confidence intervals. And the last one that I'm going to show you is 2 inactivated plus 2 mRNA, and again is close to 75 to 80% with a bit more uncertainty because of the numbers, but this is because of the number in the numerator. So the number of cases are so few that even 1 or 2 cases of ICU admission look bad, but that's for the public health perspective, is not important. So that's the population level cohort that we have, and the summary is that 4th doses work at least until 6 months after having received the booster. For most of the people, there are people that have less follow-up, but in general, people is at least 6 months after the booster. And that's a good it's good news, and there are no signs of a significant decline of the effectiveness. So we think that we are not in a rush, and we don't need to start vaccinating with a 5th dose anytime soon, at least for everyone. And this is a smaller cohort that we have in a very small town, 40,000 people town in central Chile. We selected a cohort of around 5,000 people in which we do nasal swabs for PCR every 2 weeks, no matter whether you have or not have symptoms, whether you are a contact or not. So this is people living their normal lives like community dwelling adults that we test every 2 weeks, and we measure outcomes and of course exposures. And so this is a very fine and elegant study that we've been following for almost a year. So all the data that comes from here also is just during the Omicron period. And when we analyzed infection and COVID in this group, we see that a second booster, which is mostly again mRNA booster on top of any background, which is mostly 2 or 3 mRNA, 2 or 3 inactivated vaccines, we see an effectiveness against infection. So having a PCR that's positive of 75%, and against COVID, which is symptoms, and a PCR positive, which is almost 77%. So I'm showing you this because even using original old vaccines, the 4th dose seems to be super effective against light outcomes like infection and COVID, which is something that's going to be super, I don't know, important to see whether new vaccines can increase this effectiveness, and maybe we're going to come back to a period in which we are expecting to stop transmission, or at least decrease transmission. So that's the final one. So the conclusion is that homologous and heterologous vaccine booster provide high and durable protection against severe COVID. This is especially important for the fourth dose or second booster. Against infection may be higher than expected based on the last slide that I showed you. Yearly vaccination seems feasible. We're actually in Chile going in that direction. We are going to start using bivalent vaccines very soon, but thinking about yearly vaccination and being able to monitoring V in real life settings should continue in the long run. It's going to be a challenge when COVID is not, when people start to think about the other diseases. So we have to be able to make the case to continue to monitoring vaccine effectiveness. Thank you.
Dr. Araos. Dr. Araos, thank you very much for this very comprehensive, very complicated review, whether we need vaccination or not, or does it work or not. And obviously, as you said in your conclusion slide, heterologous or homologous, it works and looks like we need a booster as long as the pandemic is going on. But the question is, what vaccine now? I mean, are we going to go on with the original ones, or do we need really the booster, the variant vaccines? And what are those variant vaccines? We are going to listen to the advances in that field from the first hands, from the ones who are developing the vaccines actually. And thank you very much again. And I forgot to announce that to our dear colleagues. If you have questions, you already realized that, but if you want to ask questions, well, there'll be a special section on the right side of your screen. You can write there. All the questions must be written down, and we will answer all the questions at the end of the last presentation. So the next speaker will be Dr. Eric Gao. Eric is the Medical Affairs Director, Sinovac Biotech Limited, China. Prior to that, he was the China lead of epidemiology and health economics at Sanofi Pasteur and the medical science team led at Pfizer Vaccine. Before joining the industry, he was the director of the Office for Public Hazard Emergency Response and the senior medical epidemiologist at China CDC for over 10 years. And he is involved in the developing of the inactivated coronavirus from the day one. And now he will explain us what are they doing there. Yes, Eric, please.
Thank you for the introduction, Professor Serhat. So I'm very honored to have this opportunity to share some evidence or update from Sinovac. So first, this is a graph that all we are familiar with. But what I want to highlight is the graph on the right, because just in the past few months, in some areas in Asia like Hong Kong, Taiwan, and also some countries like Japan and South Korea, there are some large outbreaks of COVID-19. So, and also we sadly see many deaths caused by the Omicron outbreak. So, although we see a decrease of confirmed cases and deaths from the left graph, but different countries and the region around the world have different situation. If we take an example, take Japan as an example, or Taiwan, Hong Kong as an example, there's still efforts that we need to fight against this pandemic. So that's first I want to highlight. And these next slides, I think you have all heard Dr. Rafa mentioned that there's a changing about the circulating or predominant strain around the world. But if we take a further look at the graph on the left, this is the genetic distribution of SARS-CoV-2 from UK. You can see almost every 3 to 6 months, there's a changing of the predominant variant. So that highlights that we need to be prepared always for this changing. And if take a look at the right graph, it shows, although we know BA5, Omicron BA5 is a predominant strain, but there are also different situations in different countries, like in India. So that's tell us it's very important to keep close monitoring about those variants. So this is new data from United States. You can see here, just maybe in last month, there are new variants coming, particularly the Omicron BF7. It seems just in last month, so the percentage of these new variants has increased to 2%. And also there's another new variant like BE 4.6 is also increasing its percent, and even you can see a decrease of BA5. So just tell us that the new variants is coming, and so far we don't know the severity of these new variants like BF7 and BA 4.6. So just this result, this surveillance result, just tell us we should be always alert and prepared for the new variant. So what we can do for this new situation we are facing, not new, but it's always that we may facing in next I don't know how many years, but at least in next 2 or 3 years, I think. So it's very important to think about the characters of those new variants. So now we see is the result or clinical result or clinical outcome of BE4 and BE5 if compared with BE1. So this is a result or clinical study from South Africa at the very beginning of the Omicron BE5 outbreak. So the results show that the disease severity was similar among those COVID-19 cases in BE4 or BE5 if compared with BE1. So especially for those people who have been infected or have been vaccinated. So whether you are infected before or you are vaccinated, both of them can help you to prevent from the new virus, from especially for those severe illness. So just this study, because it's very very timely and tell us that how important for vaccination. And also, if we take a look at those evidence from like inactivated COVID-19 vaccine, here this is a result for
Arkadaşlar ne oldu?
Yeah, okay. So this is the result to show the kernel 2 dose after 2 doses of coronavac. I mean, if you give both a dose among adult, so the antibody level or the neutralizing antibody level did increase among adults aged 18 to 59 years old, and also for elderly people 60 or 60 over. You can see here as this red arrow shows, there's a sharp increase about antibody levels to help to prevent infection of SARS-CoV-2 virus. And also this graph shows that the immunogenicity persistence after both doses. So here you can see the the green line shows the antibody GMT after both dose. So here just shows that even after 6 months, here you can see the level of GMT. If you give if you have take both of those, it's still higher the peak of 2 doses of coronavac. That means for if we're talking about immunogenicity, both doses can give a high antibody level if compared to 2 doses of coronavac. And also for this booster dose, we have a result from phase 2 study to show that a booster dose can also generate or induce a high immunogenicity among children and adolescents. On the left, you can see the result if we have a 10 months interval between the second dose and the booster dose. It still can give a higher increase of neutralizing antibody. Here you can see we call that the purple bar here. You can see a sharp increase. And also, if you take a longer interval, like 12 months between the second dose and second dose, you can also see an increase after a booster dose. So the circulation rates of neutralizing antibody in different groups were 100%, and the neutralizing antibody increased more than 30-fold. So this is a very good result when we talking about the booster dose against original virus or original strain. But how about the Omicron strain? So these 2 graphs shows that after both doses among children and adolescents, there is a higher neutralizing antibody level against Omicron strain. The circulation of neutralizing antibody were 90% and 91% for lower or middle dosage. When we talk about middle dosage, is standard dosage or coronavac, respectively. For children between 3 to 5 years old age group, the positive rate of neutralizing antibody was 100%. And also there indicates that there is a good cross-neutralizing against Omicron strain after a booster dose of coronavac among children between 3 to 15 years old. And also we have other immunological studies in Chile shows that after both dose of immunization, the seropositive rate of Omicron strain was over 75%, and the seropositive rate of, and also for Delta strain was over 90%. So not only just humoral immunogenicity, but also cellular immunogenicity. The neutralizing antibody and also SARS coronavirus 2 specific T cell induced by the booster dose showed activity against VOC of Delta and Omicron. So this is not just antibodies, but also cellular immune response. Both are important for the vaccines to prevent infection and also severe illness. So all those data coming from immunological studies. But when we're talking about the protection effect, just as Dr. Rafael mentioned, it's very important to test or to evaluate the effectiveness in the real world, or we call that post-marketing study. Most of them from phase 4 study, including the data from surveillance to assess the trend of disease. So next, I will share some evidence of both those of coronavac in the real world. So here, this is a very important study in Hong Kong during the Omicron outbreak, because this study shows that inactivated COVID-19 vaccine did prevent severe illness of Omicron variant. So it can prevent. So here on the left, you can see for the severe cases, both three doses or both doses of CoronaVac has a VE as 97, more than 97% among elderly people 60 and over to against a severe disease. And for adults between 20 to 59 years old, the VE, I mean, after both doses of CoronaVac is is more than 98%. And for death, the VE of both doses of CoronaVac or three doses of CoronaVac is among elderly people is about 98%. And for people between 20 to 59, we have the result of two doses because there's no deaths reported. So this is more than 94%. So this is a very important study because in Hong Kong area, we both have inactivated COVID-19 vaccine, but also mRNA vaccine. So the result, the table on the right shows that there are similar VE between inactivated COVID-19 vaccine and mRNA vaccine, particularly when we're talking about the VE against severe cases and deaths. So this is a very important study conducted during Omicron outbreak in Hong Kong outbreak. It shows that with both doses of CoronaVac, it can prevent severe illness caused by SARS-CoV-2 variants. So this study is conducted based on a cohort of Hong Kong citizens of 7.3 million adults with 12 million doses. So here, this is a very important evidence to show that both doses of inactivated COVID-19 vaccine did prevent severe illness. And also, the previous study I mentioned is among adults. So another study conducted among pediatric or among children between 3 to 18 years old, also in Hong Kong during the same Omicron BA2 outbreak. So the study, because the study period is between January 1st to April this year, so it's basically the Omicron as a predominant strain or circulating in the community. So the results shows that with three doses or both doses of CoronaVac, the VE against symptomatic COVID-19 and also the infection increased from 55%, which estimated for two doses of CoronaVac, to 92%, which indicates that three doses of CoronaVac has a very good VE vaccine effectiveness against infection or symptomatic COVID-19 cases. And also because this study not just covered inactivated COVID-19 vaccine, but also mRNA vaccine. So the VE of CoronaVac, means the inactivated COVID-19, is comparable to the effectiveness of mRNA vaccine with the same dose. So that means the same environmental. When we use two doses of CoronaVac or three doses of VAC, so it has a similar VE, just like mRNA vaccines. So this is a very important message we got from real world study. So they tell us that two doses of CoronaVac or three doses of CoronaVac did prevent the infection. And also another study also come from Hong Kong, from the same population. This study focused on the hospitalization, or we can call severe illness of Omicron. So this study is also between January the 21st to 19th of April this year. And the result shows that 2-dose CoronaVac has a VE against hospitalization. If we focus on 3 to 11 years old, it's more than 86%. If we focus on 12 to 18 years old, the VE against hospitalization is more than 90%. This is talking about the hospitalization. The VE against moderate to severe disease among children between 3 to 18 years old is more than 95%. So this is 2-dose of CoronaVac. If you take both dose or 3-dose of CoronaVac, according to the study, there's no hospitalization or no moderate to severe disease reported among 3 to 18 years old. This means that 3-dose of CoronaVac or with both dose of CoronaVac, it can prevent severe illness of Omicron, like hospitalization or moderate to serious disease. And also in this study or this analysis, it also shows that CoronaVac indicates a comparable VE to mRNA vaccine. So this is just numbers, the result from real world, just as Dr. Rafael mentioned in those studies in Chile. And this is just discussed by Rafael. I'm not repeat it, but just highlight that in this study in Chile, it also shows that with both doses of CoronaVac or 3-dose of CoronaVac, it still has a very good VE vaccine effectiveness against laboratory confirmed COVID-19. And for severe illness like hospitalization or ICU or even death, the vaccine effectiveness is more than 85%. So this study just shows that high effectiveness with homologous CoronaVac and heterologous dose schedule in preventing COVID-19 and related clinical outcomes. And the last case or last study I want to share is a study or outbreak investigation based on based on an Omicron outbreak in Tianjin, China. So during this outbreak, children or the people, not children, adult people and children who received 3 doses of inactivated COVID-19 vaccine or who get both doses of inactivated COVID-19 vaccine can reduce the disease severity of Omicron BA.1 infection. So this is very important real evidence in China, from China. And also in this study shows that the children or the adults who get both doses of CoronaVac have a lower ICU admission rate and a shorter period of hospitalization and recovery. So this is evidence from China. And also not just the VE or immunogenicity of both doses, but also some results about the safety profile. So this just shows some results based on the mass vaccination among children between 3 to 17 years old. It indicates that CoronaVac have very good safety profile because most of the AEFI are common adverse reaction, and most of them are non-serious AEFI. This just result to show that to back up the or to echo the evidence shows by Dr. Rafael about the both doses of CoronaVac or inactivated COVID-19 can effectively prevent infection and also severe illness. So far, there are about 62 EUAs or condition approval are granted to CoronaVac, and over 2.9 billion doses globally delivered for CoronaVac. So this is the basis for clinical trials and real-world studies of CoronaVac. Not just those, but also both dose and a 4th dose and a 5th dose of CoronaVac. So this is a basis, and we believe that in next few months we will have more data evidence of both dose of CoronaVac. And also, so far at Sinovac, we also are developing new COVID-19 vaccines. For example, we are developing Omicron-specific COVID-19 vaccines. We have clinical trials are going in Jiangsu and Hong Kong. And we also have a traveling COVID-19 vaccines, which covers ancestral Delta and Omicron strain. This clinical trial will start soon in Chile this year. And also about regarding the reaction. So in China, in mainland China, so far 18 people 18 years old and over should get first boost dose. You can call that third dose 6 months after second dose. And in Hong Kong area, for general population, like people aged 18 years and over, they should get a second booster dose because it may be fourth dose. And for children between 6 months to 7 years old, they are getting their first booster dose. That's for 3 or third dose. And for some specific population like immune compromised children, 60 years old and above, and also above, they should get a second booster dose. That means both dose. And I know so far there are different maybe opinions from WHO, ECDC, or US CDC. We're talking about both dose or single booster dose. So we think that it's necessary for the first dose and second booster dose, first booster dose or second booster dose. If we if we think about the situation we are facing for Omicron BA.5, and also thinking about the waning immune response for the current COVID-19 vaccines. So it's necessary to take a first booster dose and second booster dose. For the seasonal booster dose or yearly or annually booster dose, we think at least we should think about the people with high risk, like elderly people, pregnant women, and some people who have high risk, like they have some specific health condition or weakened immune system. Also for some people who may live with high risk population people, and also some people in the front line to fight against the pandemic, like health and social care workers. And in summary, we think that the pandemic is still a public health challenge for people in many countries and regions. Although globally we see a decrease on death numbers or even confirmed cases. And new variants of SARS-CoV-2 are coming, and there's still some uncertainty about their severity. So we still need to keep close on monitoring on those new variants. And inactivated COVID-19 vaccine boosters can induce robust immunogenicity and can effectively prevent severe illness of SARS-CoV-2 variants, both in clinical trials but also in real-world studies. And a booster strategy should be developed according to the local situation, but it is necessary regarding the immune waning of current COVID-19 vaccine and the changing of SARS-CoV-2 virus. And a seasonal booster strategy may be considered for the high-risk population or an specific population, which should be always put avoiding severe illness of COVID-19 as a priority goal. So this is what I want to share. Thank you very much. And back to you, Dr. Serhat.
Thank you, Eric. Thank you for the very detailed presentation. I understand inactivated virus looks like quite effective against the new variants with the booster dose, but you're at the stage of developing new vaccines, including the ancestral one, Delta, and the new variants. But the problem here will you be able to catch the latest variant to develop the vaccine, because it's always running forward. Thank you again. And we are going on with the next speaker, and Sultan Abduljawad. Sultan Abduljawad is a PhD in clinical medicine from the University of Oxford, developing vaccines. And he is an infectious disease immunologist and vaccine scientist. And most recent research was focused on the COVID-19, running one of the first studies to extensively characterize the immunophenotype of SARS-CoV-2 and the efficacy of the COVID-19 vaccines in cancer patients. At the moment, he is an external consultant and scientific advisor to BioNTech, Germany, and he will talk about the progress of mRNA vaccines. Please, Sultan.
Thank you very much for your kind introduction, and thank you so much for having me here. I'm very excited to speak to you about our bivalent vaccines. So as I was introduced, I'm Sultan Abduljawad, and I'm a external consultant for the Global Medical Affairs team. I'm a scientific advisor. As you mentioned, I am an external consultant, so this is a disclaimer about me. So what I'll be talking to you about here is the BNT162b2 original vaccine development today, just give you a bit of background to it, and then the evolution of SARS-CoV and the variants of concern, and then the vaccine immunogenicity against Omicron and its sublineages, and then the bivalent vaccines we have at the moment that are coming out in the vaccine effectiveness against these sublineages. So going back just to the beginning, talking about the BNT162b2 vaccine, which is the original vaccine that was released last year. So this is Project Lightspeed. That was basically the development of that vaccine. And the aim here was the development of an effective and well-tolerated vaccine following global scientific, regulatory, and ethical standards. And this was done within 10 months. And just to kind of give you a very brief kind of timeline from start to finish. So when we think of when the pandemic started and then when the sequence was first made public, January 12, 2020, within 2 weeks, we already started the preclinical studies with 20 vaccine candidates using different antigens, for example, full spike or receptor binding domain, and also using different RNA platforms like the modified RNA or the self-amplifying RNAs. And these all entered preclinical testing at that stage. And then after that, we started actually collaborations, two major collaborations. One was Fosun Pharma for the regions of mainland China, Taiwan, Hong Kong, and Macau, and also a global collaboration with Pfizer. These were done in March. And then by April that year, the first phase 1/2 clinical trial started in Germany, and by May, it started in the US. And at this point, from the preclinical study, we had 4 vaccine candidates to enter the clinical testing of phase 1 and 2. And this is to look at the safety here and immunogenicity. By July, we already applied for the FDA Fast Track designation. And by the end of July, the pivotal phase 2 preclinical trial for the BNT162b2, which was the lead mRNA vaccine candidate at that point chosen, due to the safety profile for it and immunogenicity, had started. And that was at the time the largest COVID-19 vaccine trial in over 44,000 subjects. Now, as the trial was ongoing from July, by October, we already started the rolling submissions with different regulators around the world. So it was EMA, Canada, UK, Singapore, New Zealand, and other countries. And by mid-November, the phase 3 clinical trial met all the primary efficacy endpoints. And then we started the rollout by the end of that year. So you can see here, from start to finish how, the process and and what it involved. And at the moment, we are in over 170 countries around the world with different regulatory approval, and now it's authorized in 6 months and over. And it has also been expanded for boosters in 12 years plus. And also for, of course, enforced boosters have been implemented in high-risk cohorts in selected countries. So this is the background what we had till this point. Now, as already been discussed quite extensively here, but I'll just touch upon it a bit, is, SARS-CoV-2, as we know, has been evolving, and there's been quite a lot of different variants over the last couple of years. Now, some of the mechanisms that can facilitate these mutations include intra-host evolution due to immune system impairment, or suboptimal neutralization antibody treatment, or from viral reservoirs in unvaccinated or immunocompromised individuals. Now, resistant strains emerge regardless of transmission rates, but for the strains to become established, they do require high transmissions. And high infections within populations increases the probability of gain-of-function mutations because they actually require a high number of viral replication events. And as population immunity drives the selection and the pressure of the mutation that evades antibody neutralization. So variants of concern, they can emerge from any prior lineage, regardless of the current prevailing strain, although most variants actually will emerge from the most highly transmissible variants. And just looking here at the right side for on the phylogenetic tree, it's just to kind of highlight, as already been highlighted in the previous talk, how divergent the Omicron variant is from the different variants, that it's almost a whole new subgroup. And also when you look at the number of mutations, so the Alpha variant has the fewest number of mutations, whilst the Omicron has 30 mutations. And this kind of suggests that Omicron was circulating for possibly a long time before it was actually discovered. And if we move on looking at the epidemiology of the different waves, so now Omicron, as we know, is becoming the most prevalent globally, specifically now BA.4/5 at the moment. But if you look at the timescales of the different variants of concerns, for example, the Alpha, the Delta, there was kind of a 6 months to 1 year timescale of the rise of that kind of variant and then kind of going down. But at the moment, that's kind of shifted with Omicron, and we're seeing this kind of 3 to 4 months emergence of variants, and then this other variant coming and overtaking the previous variant. And this is due to the really high transmission of Omicron and also the evasion we're seeing with Omicron. And if we go a bit more specific and looking at the BA.4/5 subvariant, so they demonstrate actually a greater immune escape than both BA.1 and BA.2. And they're actually resisting neutralization antibodies from Omicron-naive triple vaccinated individuals more than we saw with BA.1 and BA.2. And also, looking here at the phylogenetic tree, you can also note that the BA.4/5, the distance from it to BA.1 is actually is kind of a similar distance you saw originally with the Delta and the Wuhan strain. So although they're in the same subgroup, they're quite antigenetically distant from each other. And there's quite a few mutations that enable immune escape, but some of the ones to highlight are these 3 mutations in the receptor binding domain that are found in the BA.4/5 that have been highlighted and shown to enable this agent of neutralization. But also we're seeing the clinical manifestation of these new variants of concern actually different. For example, they're targeting the lower respiratory tract, and they're potentially more pathogenic than BA.1 and 2. And also to highlight here the distance, we're seeing also with Omicron BA.1 breakthrough infections in vaccinated individuals, they demonstrate actually very limited cross-neutralization activity against BA.4 and 5 when you look at durability of response. And again, this was also noted within the vaccines, and this was also highlighted in Professor Rafael's first talk, and we see waning of vaccine effectiveness against all the Omicrons. We first noted it with the BA.1 and BA.2, and now, of course, we're still seeing that with BA.4 and 5, especially after 3 or 4 months. And more specifically, with infection, we're seeing, quite a reduction after 3-4 months of effectiveness of the vaccines against symptomatic infection. However, against severe disease and hospitalization, there is also a waning going on after 3-4 months, but it's less severe, and there is still a high kind of vaccine effectiveness. But this highlights basically there is a waning here, and that we do need some, better protection against these variants of concern, especially since they're very divergent antigenically. And the increased immune escape properties that are demonstrated by these by the SARS-CoV-2 variant highlights the need for basically a variant-adapted vaccine, and more specifically an Omicron variant-adapted vaccine. Now, when you're boosting with the original vaccine, you are recalling your old memory, and which are going further affinity maturation and somatic hypermutations, and you're expanding the breadth, but it's not expanded, but there is still a lack of breadth in the response to these very divergent variants of concern. And that's why we were so early in the talk that the original vaccines were very effective against alpha, beta, and delta, and we're seeing it less so with Omicron due to the antigenic distance. However, if you boost with an Omicron variant-adapted vaccine, what we may result in is you're going to have activation of new naive B cells, production of higher neutralization titers against the new antigen in associated sublineages that may arise from these antigens or are more closely genetically linked to these variants of concern. You're also going to have an increase in the breadth of the epitopes targeted by the expanded B and T cell responses. And you also have a reactivation of the performed memory B cells, but also you'll have a formation of new memory B cell repertoires, which will also expand your antibody depths. And also this will offer potential cross-neutralization of similar new variants of concern, which in turn will give you more durable protection. So here, variant-adapted vaccines will improve the cross-neutralization and will expand your memory B cell formation against conserved epitopes and new epitopes and elicit increased neutralization titers against Omicron and associated sublineages. And one of the reasons why, or the mechanism why, is because of the formation of this new germinal center, appropriate germinal center, and the result of this will increase the neutralization titer, will increase the formation of new B cells, new memory B cells, and production of antibodies with higher avidity and cross-neutralization ability. And this will in turn, of course, reduce infection rates against this new antigen and any aligned sublineages that may arise. So by increasing the breadth of memory B cells, it will lead to a faster, effective response to the variants of concern with a similar genotype. And also, immune responses against new variants may not be hindered by antigenic or immune imprinting because the new germinal centers could be generated, leading to breadth of antibodies capable of discerning these new variants. So here, I'm going to jump into the trial for the BA1 adapted vaccine, and this is the trial that was initiated last year. And what you see here is, so people who've had the three vaccines of the original vaccine, then we had three different arms. Either they got a fourth booster with the original vaccine at two different doses at 30 or 60 microgram. Then we also tested out a monovalent BA1 vaccine. So this only the BA1 Omicron is high and low, 30 and 60 microgram. And then we have the bivalent BNT 162b2 vaccine, which is which included the original variant and the BA1 in at half. So at 30 microgram, you have 50 microgram of the original, 50 microgram of the BA1, and at 60, you had 30 and 30. And I'm going to focus now just on the bivalent one, since that is what's now being rolled out. But here you have the neutralization titers against Omicron BA1 from all vaccines. And if I point your attention just to this one here, so this is the bivalent 30 microgram, and this is here the original vaccine, and we're seeing, and you're seeing here the neutralization, and you can see there is an increase in the neutralization titers, and it actually met the simple superiority criterion. And here you can see the GMR of 1.56. But what's also important is that we retain the immune responses against the original variant in case new variants arise from that. And this is what you see here, that responses against the reference strain, the Wuhan reference strain. This is here the original vaccine, and this is the GMR. And we can see here with the bivalent Omicron vaccine, we maintain a superior response also against the original vaccine. And then when we look at the different subvariants here of Omicron, so the BA1 adapted vaccine as a fourth dose did elicit improved BA1 neutralization responses, but it was more modest against the BA4-5. BA1 here is in dark green, and BA4-5 responses are in light green. And we can see here the response against BA1 was, and that's the GMR, and then there is a drop against BA4-5. So it's still producing better responses than the monovalent vaccines, However, it is lower than the BA1 vaccine. And when we looked at the safety profile in the reactogenicity, so it showed a similar local reaction and systemic event profile as the prototype vaccine, which is really important. This was in individuals 18 to 55, and also in individuals above the age of 55. The modified adapted vaccines also had a similar local reaction and systemic event profile with no new signals detected. And even at the high dose, there was only mild to moderate injection site pain, fatigue, and muscle pain, which were common compared to the 30 microgram dose. And if we go back looking at the murine studies of the Omicron BA1 monovalent and the bivalent vaccine, again, it shows the same thing where you have an increase in the immune response against BA1 and there is a reduction against BA.4/5. But however, you still note, so looking here at the bivalent vaccine, which has the wild type and the Omicron BA1, and then looking at the original vaccine, so this is the responses against the Wuhan strain, the reference strain. These are the responses against BA1, and this is the response against BA.4 and 5. And you can see here with the bivalent vaccine, we maintain the responses against the original, and then we actually have quite a significant increase in the responses against BA1, and also quite a significant increase in responses against BA.4/5, although it is lower than BA1. So there are more neutralizing antibodies being produced against the subvariants of Omicron than there was with the original vaccine. And we've also started with our second bivalent vaccine, which is already being rolled out, which is the BA.4 and 5 adapted booster vaccine. This is now currently just started in clinical studies, but this is here the results from the murine study. And what we do see is that the BA.4/5 adapted vaccine substantially increases the Omicron neutralization responses against all Omicron variants, actually. And this is here the bivalent BA.4/5 vaccine, and this is here the original vaccine and the monovalent BA1 vaccine. And if you look at the responses, for example, to the wild type, it still maintains responses to the wild type, whilst the responses against BA1, BA2, BA2.1, and BA.4/5 are all substantially higher than the original vaccine. And against the monovalent vaccine. So what we're seeing here is good cross-protection and cross-neutralization across the different variants. And that's why we think it's the way to go. So just in summary, what we see here, so robust vaccine effectiveness data were generated globally to demonstrate that a booster dose of the BNT162b2 increases protection against symptomatic disease and severe disease caused by both Omicron BA1 and 2. However, there is a waning in protection against symptomatic disease after a few months, and the durability of this COVID vaccines greater than 6 months post-booster against severe disease remains unknown, especially against these newer variants of concern with enhanced immune escape mechanism. And SARS-CoV-2 epidemiology continues to change rapidly with a new variant of concern arising every few months, and those with greater transmissibility and enhanced immune escape are displacing these prior prevalent lineages. So bivalent Omicron vaccines will provide a wider breadth of protection against previous VOX, Omicron sublineages, and antigenically aligned lineages of SARS-CoV-2 than the original vaccine. And the safety of the booster dose of the Comirnaty original Omicron BA.4/5 was inferred from the data of the booster dose of the Omicron BA1 adapted vaccine, as well as for the booster dose of the Comirnaty original vaccine. So now the bivalent vaccines, both BA1 and BA.4/5, are being reviewed for recommendations globally, and they have been authorized at the moment by the US FDA and EMA in Europe and other regulatory bodies for subjects 12 years of age and over. And the bivalent vaccines will be implemented in prioritized populations according to local country guidance prior to expanded rollout to the broader population groups. And thank you very much.
Thank you, dear Dr. Abduljawad. Thank you for this brilliant presentation. So the mRNA vaccines obviously are easier to adapt to new variants, and see the BA1, and BA.4/5 is already ready. Very good. And dear colleagues, we are going on with the last, of course, not the least speaker, the third type of vaccine, vector vaccines, will be presented by Dr. Bárbara Emoingt Furtado. Dr. Bárbara Furtado graduated as a medical doctor at the Federal University of Rio de Janeiro, Brazil, and specialized in pediatrics and pediatric oncohematology, again in Rio de Janeiro. She joined the pharmaceutical industry in 2012, and since 2016, she has been decided to the area of the medical affairs for vaccination. During this period, she has participated in the publication of 4 articles and a book chapter, and she is currently the Associate Medical Director for Vaccines and Immunotherapeutics in AstraZeneca, UK. Please, Bárbara.
Thank you, Dr. Serhat, and I want to thank to all the organizing committee for the kind invitation. As already presented, my name is Bárbara Furtado. I am the International Associate Medical Director for Vaccines and Immunotherapy in AstraZeneca. I'm delighted to be here today. This is my conflict of interest. Today, I'm going to present an overview of the adenovirus vector vaccines. I want to show you a recent publication discussing the vaccine effectiveness of the booster dose for COVID-19. And on top of all the information already presented by my colleagues, raise some discussions about the vaccine future. So adenoviral vector vaccines are nothing new to the scientific community. In the matter of fact, the concept of the viral vector was introduced decades ago in 1972. And in the early 80s, the first vaccine virus-based vector was described. From then on, more than 40 vectors are used as viral vectors for vaccines and gene therapy. And the adenovirus is the most common vector used nowadays for vaccines design. And why is that? The adenovirus are non-enveloped DNA virus with double-strand DNA gene. And they present many benefits on top of other viruses, as they are easily genetic manipulated. They can be non-replicating through the deletion of E1 domain. And this is a particularly important information regarding the most vulnerable population, including the immune-suppressed patients. They accept large transgene DNA inserters, and they can generate, as we already know by all the COVID-19 vaccines studies, they can generate a robust specific T and B cell response. So how do these vaccines work? I like to start this explanation by referring to a well-known historic fact is the Trojan horse. The history say, as you already know, that the Trojan horse was a wooden horse used by Greeks during the Trojan War to enter in the city of Troy and win the war. And what is the relation between this and the adenoviral vector vaccine? The adenovirus act like a Trojan horse. Its structure is used to penetrate the cell, and in the inside of the adenovirus, we have what will allow us to win the war, the warriors. So it is the SARS-CoV-2 S protein mRNA. Once injected, and let me show here, once injected, the adenovirus will fuse with the host cell, and this fusion allows the ES protein to be inside the cell. And this inside the cell, it's the protein, the mRNA as protein will reach the cell nucleus. The host cell so replicates and express the S protein to the cell surface, permitting that the host immune system recognize the S protein, and then B cells as neutralizing antibodies and T cells response is triggered. The memory B cells and T cells are formed, and this is very important when we are talking about the long response for those vaccines, priming the immune system to attack the SARS-CoV-2 during the subsequent infections in virus. But do those vaccines work? We could see during 2021 that those vaccines worked against SARS-CoV-2 and COVID-19. It's clear now that the adenoviral vector vaccines were an important tool in the war against COVID-19 pandemics. But during the pandemics and moving from pandemic to endemic state, some questions were raised. So how are current vaccines faring against Omicron, and are new vaccines required to address the variants of concern? Are all vaccines equally effective as a booster, or are some booster schedules more effective than others? And we are going to address a little bit about that. And the $1 million questions: do we need to keep receiving booster dose, and how effective are those booster doses? So to address some of those questions, I want to share with you the results of this expert review of global real-world data on COVID-19 booster dose effectiveness and safety during the Omicron dominant phase of pandemic. This expert review includes 52 studies worldwide. And this is the top-level overview of this project, that we include 52 studies. The expert review evaluates the outcomes as symptomatic infection, hospitalization, and death. And we are going through the key findings of this study. Just to point out the characteristics of these studies, I want to share with you that both mRNA vaccines were included. Studies with both or the most used adenoviral vector vaccines were included for AstraZeneca and Janssen, and CoronaVac as the inactivated virus vaccine was included as well. We have different study designs, but all the study designs need to have a control arm. And this control arm could be unvaccinated population or partially vaccinated population. And the outcomes that were included were severe COVID-19 disease, symptomatic disease, and death. So the key findings of this study. So here we can see the homologous booster dose schedule for the different vaccines, and we are going to talk a little bit more the comparison about the homologous booster schedule and the heterologous booster schedule. So for these results, we can see that all the homologous booster schedules find out a very good vaccine effectiveness against severe disease. And for the purpose of this meeting, I'm just going to address severe disease and death, and we are not going to discuss symptomatic disease in this presentation, but we can discuss further in the Q&A. So what we can see is equally response against severe disease for all the vaccines. I just want to address a little bit here my theme that the adenoviral vector vaccines, that we can see here that almost all of them above 80%, but Janssen vaccine were a little lower than 80%, and this may be related to two things. Maybe we have fewer studies using homologous booster as compared to the mRNA vaccines. And we have to have in mind that for Janssen vaccine, 2 doses only, we consider the primary schedule vaccination with 1 dose. And here, as we are talking about the 3rd dose, just 1 dose for the booster dose. And this may have some impact in those findings here to this vaccine effectiveness a little lower than the other vaccines. But in the end, we have a pretty good response for all the vaccines regarding severe disease and death. And remember, this was during Omicron. For the heterologous booster, what we are seeing here is the same. A very good response for all the vaccines with all the schemes that I'm going to go through all of them for, again, severe disease and death following the 3 doses. Once more, I want to highlight that here, the adenoviral vaccine from Janssen, we had a lower vaccine effectiveness, but it's important to understand that we have fewer studies for this vaccine. And once more, we are considering the primary schedule vaccination, just 1 dose for Janssen. So here we have Johnson vaccine followed by a Pfizer booster. Here we have the AstraZeneca vaccine. So the AstraZeneca followed by Pfizer and AstraZeneca followed by Moderna vaccine, reaching out more than 85% of vaccine effectiveness. Here we have Pfizer vaccine followed by a Moderna booster. And here, different schemes from Pfizer and Moderna that were put together, but we can see that they reach a very good response. As Dr. Rafael and Dr. Eric already shown, CoronaVac followed by a Pfizer booster reach out a very good vaccine effectiveness, and the same for Moderna followed by Pfizer booster. So what we can see here is the booster after any primary schedules provide equivalent protection against Omicron-related severe disease and death. And the protection against severe disease and death is comparable for heterologous and homologous booster schedule. And this is a very important discussion that we were having during 2021 and 2022, if we should go to a heterologous booster or we can maintain our population in an homologous vaccination schedule. And this work showed us that they are equally important to protect against severe disease and death. So the highlights of this paper, it's the equivalent protection against severe Omicron outcomes following the 3 vaccines booster was observed with both adenoviral vector vaccines and mRNA vaccines with no significant waning over 90 days. The consistent effectiveness of current vaccines against all strains include Omicron. This is very important because we are seeing low vaccination coverage for 3rd dose and 4th dose, mainly in the low and middle income countries. And the vaccination program should focus on expand coverage of the populations with the already available booster shots. Of course, the new generation of vaccines are very important, but we cannot wait for them to come to all the countries. The level of protection after the booster is high, regardless of whether the booster shots were the homologous or heterologous from the primary vaccination series received. And this data are supported by a recent real-world 1st and 2nd booster dose data from Thailand, further highlighting the importance of improving booster dose coverage against Omicron. I didn't brought this data from Thailand, but what this paper showed to us is the 4th dose improve the neutralizing antibody against the Omicron variant. And they can reach a very good vaccine effectiveness with this 4th dose booster for all the vaccines. So looking ahead, the future of COVID-19 vaccination, what the future looks like. And of course, it's not a crystal ball. I want to share with you some thoughts. So maintaining a level of immunity across populations via regular booster will likely be necessary to protect the most vulnerable population and reduce the impact on economies and healthcare system. What we need to address here is who is going to receive regular booster dose, just the vulnerable population or the whole population, and will be annually doses looks like could be that way, or we are going to need more frequent boosters. This will depend of some discussion, including the development of next generation vaccines that offer timely additional value beyond existing vaccines, and then can represent the next stage of protection against COVID-19. But to answer all this information, further research on SARS-CoV-2 evolution are necessary, as correlates of protection is still needed. So some vaccines already been developed, some still under development. But when we are going to discuss the next generation of vaccines for COVID-19, it's important to have in mind that these new vaccines need to provide greater and more durable protection against severe disease and death than the current vaccines. And of course, may take some time for us to discover if this is going to be true, since we are starting to vaccinate with new variant vaccines. They need to provide broader protections against future variants that are more antigenically distinct from the index fire, the Wuhan strain. And of course, once more, we need those new variants to raise and to understand if those new vaccines are going to be effective to them, against them. And they need to prevent spread to vulnerable population and reduce the risk of new variants emerging. And something that's a desired benefit that we saw in the beginning of the pandemics, but we are not seeing anymore, it's improved protection against symptomatic illness and maybe transmission. So the monovalent vaccines, and all of them until now were monovalent. Several vaccines are in development that target the spike protein of one variant only. And talking about the future, I just want to point out that AstraZeneca already have a study, the AZD2816, to address this need. We have a study that was made with beta variant in 2021, and the study came out with a positive result showing us that it's possible to change the vaccine, and immunogenically, we are talking about an improve against, for example, the beta variant. That was the study, and we can maintain the gains that we have against the alpha variant and Wuhan variant as well. For bivalent vaccines, I think that we already addressed something in the last presentation, and those vaccines target two spike proteins of one of the ancestral strain and one of Omicron variant. And beyond spike, additional parts of the viral proteome are also being targeted as an antigen. So what we have now for the next generation, some of them data already available across multiple variant vaccines, like the mRNA-based vaccines, Pfizer and Moderna variant vaccines are now approved in European Union and US. We have adjuvanted recombinant protein-based vaccine, and we have new adenovirus vector-based vaccines already under development. Other platforms are under investigation, like the mRNA universal coronavirus vaccine, self-amplifying RNA vaccines, and new inactivated vaccines as well. So to summarize, the booster of adenoviral vector vaccines offer protections against the severe disease caused by variants of concern, include Omicron, and they are comparable to the other vaccines platform. The vaccination program should therefore focus on improve the booster vaccination coverage now. Several next generation vaccines are currently under development, involving both new and existing vaccine platforms, representing the next stage of protection against COVID-19. Thank you very much for the opportunity to be here today, and we can address some doubts in the Q&A. Thank you.
Bárbara, thank you very much for this very didactic lecture. I understand AstraZeneca is working on the Omicron variants as well. That's very good. Now we can go on with the questions. Thank you all, dear colleagues. We were timely finished, and we have time for the questions. First 4 questions are asked after Dr. Araos' presentation, but addressing all of you, you please feel free to answer if you need anything more to say. What are the side effects of revaccination for COVID-19? Asnain Sajat. So the more allergic, more side effects, I think he means. Yes, we can start with Rafael. Rafael, do you have an answer for that?
Yes. So we don't have an active surveillance for adverse events. So what we collect is, it's going to be, just what people notified, and we don't see any different specific adverse event related to the subsequent doses. So that's one thing. There's no like class effect of boosters. And the other thing is that we don't see more rates of severe adverse events with subsequent doses, which is something that's in line with what Israel has shown. There might be something related to reactogenicity. So people tend to say and report more local pain, but from a statistical standpoint, we haven't seen any red flags.
Thank you. Sultan, you already mentioned about that the revaccination studies, you didn't observe any serious side effects, but would you like to recommend on that one more time?
Yes, you're right. So yeah, so in the booster studies, including, so there's multiple studies. There's the original vaccine booster studies that was done in the different age groups, and then there was, of course, now the booster vaccines with the bivalent vaccines. And yes, there hasn't been any new signals detected at all in the clinical studies. And also, of course, we have the post-marketing surveillance for at least for the original vaccine that's been ongoing for the last year and so. And there hasn't been any kind of new signals detected. There's the old known side effects. But yes, so the booster itself is not causing a specific, if you want, kind of as you said, like a side effect because of the revaccination, as Rafael just said.
Okay. Eric, Bárbara, do you have any different observation than that?
Yes, I just want, not a different observation, but just want to add to, I just want to add two comments. One is specifically to the AstraZeneca vaccine. Looks like that the first dose is the more reactogenic one. So we are not seeing the studies in the real life studies or AstraZeneca studies, an increase of adverse events with the subsequent dose. But for the adenoviral vaccines, it's important to address the antibodies anti-vector. So in all these studies, we are trying to address this question if regular boosters from the with the adenovirus vaccines will generate antibodies anti-vector. So until now, we couldn't find that this is a problem, but of course, with more booster dose, we need to follow up with this question specifically to the viral vector vaccines.
Eric, anything specific to inactivated ones?
Yes, during our clinical trials of the booster dose, we didn't see serious AR or EEFI reported. So that means it's quite safe for inactivated COVID-19 vaccine as a booster dose. And for the post-marketing surveillance or monitoring for mass vaccination, so most of the AEFI from those doses are common AEFI, and most of them are non-serious AEFI. So that's what we have observed. So during the mass vaccination, inactivated COVID-19 vaccine as a booster has showed a very good safety profile.
Thank you. Another question from Dr. Serhat again. It is, he is asking, let's say someone got regular doses, including booster, and did not suffer from the infection at all. He's asking, do we need revaccination of those people as well? And I'm advancing that question. Well, let's say 3 doses, and then no disease, 3 doses, disease after that. And what is the plan of the revaccination of these two different situations? I'd like to start with you, Rafael, again.
Can you repeat the first part? For some reason, I couldn't.
The heterolog or homolog booster completed, it's finished, and asking and the patient or the person did not suffer from any disease. He is not committed disease. do we need a revaccination after, let's say, 6 months? Because we know that after 4 months, the antibodies are waning. And I'm adding another question. Let's say got the booster dose regularly and then suffered from the commuted disease 3 months after that. And shall we vaccinate that person? Either disease happened or not.
Yeah, so it's those are great questions, and I can give you my opinion. It's not a final answer, but after vaccinating people with let's say an extended primary series, so anything plus one booster, it seems that protection against severe disease lasts a good amount of time, at least 6 months. However, countries that can tell you that, like Israel or my country, maybe others, we are continuously vaccinating people until now. So it is hard for me to say what happened after 3 doses and 6 months, because we already put a fourth as a programmatic intervention. So in a way, it's hard to know what happened with those with 3 doses and not the fourth dose that was indicated. So that's something that's important. So if you think about the kinetic of the neutralizing antibodies, we know that even after a 3rd dose, there's an important drop, and that's pretty rapid. But the T cell response probably is going to last much more time. And I think if you have to decide, I would try to cover as many people as you can with 3 doses before starting to think about a 4th. And then with the 4th dose, I would start with the high risk population, and then up to new information to decide whether to vaccinate the whole population or not. We vaccinated everyone because it was a decision beforehand, but probably only having vaccinated with a 4th dose and focused on those at high risk would have been okay anyways. So that's one thing. The other question is basically hybrid immunity, which is something that's super hard to study. So it's very difficult to understand, at least from the observational perspective, how to approach that. But it's pretty reasonable that if you have a decent way for confirming the infection, let's say you PCR the subject or it has symptoms, I don't know, but you're convinced, you can consider that infection as a booster, and there are plenty of data from the at least immunogenicity that can come out of that, and several observational data saying that it is okay. I mean, so hybrid immunity, it is probably a fact in COVID, and I would consider that from a public health perspective, if you can record that, in your records and at the country level, those individuals can skip the next dose until they need to be covered by the yearly program or whatever like that. That's my position.
All right, thank you. We still have lots of questions, so I'm getting into the next one. Next one is, I think we have answered this question. Regarding revaccination in COVID-19, have you tried heterologous against homologous booster in enhancing immune response? The answer is yes. All of you almost mentioned about that. Does not make so much difference. We need booster, and it may be homologs or heterologs. I think everybody is on the same page for that. This question is also for you, Rafael. What was the rate of breakthrough infection in your study population?
So it's really hard to answer that here because we have so many groups, but it's going to be in the tables in the paper that's going to be uploaded really soon. But it's an important number. I mean, infections do occur in people with vaccines. If you have everyone covered by vaccines and by boosters, as we have in Chile, so the number of people that are vaccinated here is very high. So everyone with an infection is basically a breakthrough infection. So we do see a lot. But even when you compare effectiveness against infection, those without vaccines have significantly more events of infection, as I showed you in the last slide of this small cohort that we follow with weekly PCRs. So that's pretty clear. It's totally different, the rates of infection between those that are vaccinated with any number of vaccines less than 6 months after the last dose, from the second dose onwards, compared to those that are unvaccinated.
Okay, thank you. Dr. Gao, there are specific questions for your presentation, and the adjuvants added to the inactivated vaccine.
Oh yes, we use aluminum adjuvant as a tool to enhance the immune response. So this is a technical, classical technology used when we develop vaccines. We can think about the poliovirus or the IPV vaccine. See, we use the same technology. We use the same way to develop inactivated inactivated viral vaccines. So and also I can see another question covers the problem or the challenge when you use this inactivated vaccine. But those issues or challenges have been well controlled when we use inactivated technology. So it's no problem. You can think about that. So we use SIPV vaccine as a weapon to fight against poliovirus. Now we are close to the eradication of the poliovirus globally. So there's not a big issue if whether you use a very new technology or you use a classical technology. So both can work, and it will be confirmed by the real world study and the public health impact in the long term.
Thank you. That question was asked by Hu Wen Chen. I think he's from China. And another question to you by Dr. Isato Sar. What do you think on the problems associated with the virus inactivation process, such as virus aggregate formation, protein cross-linking, protein denaturation, and degradation? Inactivation process, what was the method you used, and what were the problems for that? Did you get the question? Eric, could you get the question? You are muted, Eric. We cannot hear you. Oh, yes.
Okay. Could you repeat that again?
What was the what were the problems associated with the virus inactivation process, such as virus aggregate formation, protein cross-linking, protein denaturation, and degradation? The inactivation process.
Yeah, yes, just as I mentioned, so it's a classical way, but the challenge or issue during the inactivate process have been very controlled. So we use the same technology to produce SIPV vaccine in Estanovac. So there's no big challenge about this problem, I think.
Okay. And Dr. Abduljawad, Why did BioNTech Pfizer not choose BA.4/5 monovalent 1 microgram vaccine, as it showed even higher antibody levels in the shown graph in red? Bernard Haas from Austria.
Thank you. So I just want to highlight here first, so that 1 microgram, this is the murine study, that's why it's that low dose. But the reason it's the bivalent vaccines and not the monovalent is not just based on the study. It's actually the consensus within the scientific community and also within the regulators that moving forward they want to move with the bivalent, not a monovalent. And this is so it's very important here by having the original. I just spoke earlier about possible new variants coming. Now we can assume maybe a new variant will arise from Omicron, but cannot be sure of that. So maybe a new variant will still arise from the original Wuhan virus or from other subvariants, variants of concern. So it was important to still include the original vaccine to maintain the immune responses against that genetic, if you want, variation there. But also here, you're enhancing it by including the new antigen. So the bivalent approach was what was agreed upon, if you want, or the consensus moving forward. Hence why we chose, we went with the bivalent BA.4/5, not the monovalent.
Okay, thank you. And the next question goes to all of the participants, all of the lecturers. Dr. Aizato Sar is wondering, there are reports that the current vaccines are inefficient in stimulating IgA secretion in mucosal cells, which may not be efficient in controlling the shedding of the viral particles in the upper respiratory tract. And what do you think about combining both local nasal vaccines? One nasal vaccine has already been approved in China, as you know, but he or she is, our colleague is asking, is it logical to combine nasal vaccine and systemic intramuscular vaccine in enhancing immunity and stopping the transmission of the virus, not only protecting the severe disease and death? Can we start with you, Bárbara?
Sure, thank you for the question. I think that first of all, it's important to address that we have plenty of information that the actual vaccines do generate IgA response, maybe not enough to stop transmission and not enough to reduce the viral load, but they do respond somehow to IgA. So I think that of course, when we are talking about respiratory virus, the transmission and the viral load, it's always a demand that we need to address. I think that look for, and we have already some studies in this going to this pathway of intranasal viral vaccine. They are important, but we need to remember that the intranasal viral vaccine, they are usually live attenuate virus. And with this come the discussion if we can use in the most vulnerable population, in the elderly and the immune suppressed. So we are not going to have it all. Probably we are going, if it's possible to have an intranasal vaccine, it's awesome. I think that's really good to have that, but maybe we are not going to use this vaccine to all. I think that's difficult to have one fit for all. If we compare to influenza, we do not have until now. And I think that we are going to find out more ways to combat the COVID-19. So I think that use both together, we do not have any I think that we do not have any information regarding use both. I think that if we have an intranasal vaccine, we're going to have EGA, but we're going to have EGG as well. So this is going to be enough to address the infection, but the injections and the inactivated vaccines that we have today are going to address for the immune suppressed and the elderly probably better than the intranasal vaccine.
Yeah, thank you. So what do you think, Eric? Is that combination possible?
I think it's an idea, but I don't know if it's possible in the real world or even in the clinical trial. So it's a way we can try, and let's see what we got from clinical trial or and also real world study.
Okay, thank you. How about you, Sultan?
I have to say, I think Barbara covered it really nicely.
All the bases, yeah.
Covered all the bases. Yeah, I don't. I at the moment I wouldn't have anything to add on to that.
Thank you. And Rafael, what do you think?
Two things. One, I endorsed Dr. Furtado's comments. I totally agree. And the second one is, it sounds like a consigno question. We're waiting to see that that results, and if you make it to the public, I will tell you how it goes in the country.
Okay. All right. Well, dear colleagues, there are more questions, but we are out of time. I can promise, I guarantee that those questions will be sent to the people who are addressed, and I'll ask you, if we send you an email, will you promise us to answer them back, because we are out of time now, and we will answer you that privately by the email. I'll send the emails to the people who are addressed, who are asked the question. Well, thank you very much. I mean, I really enjoyed every minute of that, and I have learned a lot. Still the question going on, but I understand that we all agree that we need booster dose, definitely. It's obvious. It may be homologous or heterologous. And we need revaccination about 6 months after that. And it looks like that the original vaccine is still keeping its activity, although it's going down a little, but it's still keeping activity against the new variants in the perspective of the severe disease and the death. But it may be better if we get the variant vaccines. But shall we be able to race with the race of the emerging of the variants? Can we catch them up? And we will see them by the time. And again, it looks like that in the next coming, I don't know how many years, but we will go on the perhaps seasonal revaccination with the corona. Perhaps we'll talk about the combination of influenza and corona vaccines in the coming years. We will see all together. Thank you again for joining us today and contributing with your lectures, and all the colleagues who has listened to us from 63 different countries all over the world. Good night, good evening, good morning, again, whatever you say, and you see all in the next coming meetings. Bye-bye.
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 Bye-bye.